Hello everyone, welcome to ITLS Academy. In this session, we will be talking about the second session of the topic protein therapeutics. This is your mentor, Dr. Shamaita Basu. So, as I mentioned in the previous session, that in this session we will be mainly focusing on the concept of monoclonal antibodies because they are a very important part of protein therapeutics. Okay. So before starting with monoclonal antibodies, I would just like to give you a brief introduction about antibodies. I think antibodies are something that I've already talked about in the previous sessions of this course of medical biotechnology. But anyways, so antibodies are basically naturally occurring proteins which help our body to fight against any infectious disease, and any antibody will be produced in the body only in response to an antigen, okay, not before that. So this is a typical structure of antibody, Y-shaped structure. So these are the heavy chains, these are the light chains, uh, we have the constant regions, we have the FC regions, and we have the FAB regions. Okay. Okay, now B cells will be producing antibodies. So there will be B cell antibody receptors which will be recognizing the antigens, and after recognizing those antigens, signal will go that okay, now you need to synthesize the antibodies. So the B cells will basically activate the plasma cells, and the plasma cells will go and synthesize the antibodies. That is the main pathway. So the antigens are molecules that cause an organism to generate antibodies, and the B cells activate when their antibody receptors bind with the antigen. Activated B cells differentiate into plasma cells and the memory B cells, and then these plasma cells secrete the antibodies. So basically from the B cells, the plasma cells will be formed as well as the memory B cells will be formed. And what is the function of the memory B cells? That if in future that same antigen again attacks your body, then there is no need of again sensitization of the antibodies. Directly those memory B cells will be activated and they will be producing the antibodies. So for the second time, if the same person contracts the same disease again with the same strain or the same variety of virus, then what will happen? There will be no, like within a short period of time, the body will be getting activated by producing the antibodies. Okay. So I've just given a nice diagram to make you explain. So these are basically the antibody receptors that are there, and this is a typical B cell. So this is basically how the activated B cell will look like, okay, with the receptors attached, with the antigen attached with the receptors on the B cell. So the activated B cell will just go on and start producing the plasma cell and the memory B cells, which will be containing the receptors, and as a result the plasma cells will be producing the secreted antibodies. Okay, now these are just different examples of monoclonal antibodies, which I will be talking about in the upcoming slides in more details. Okay. Now, there are multiple B cells which will be generating antibodies that bind with different regions of the antigen. Now, this is a very important concept. Now, what are those different regions known as? These different regions will be known as epitopes. So, epitopes are basically those regions where the antibodies will go and bind with the different antigens. Not different antigens, different regions of the same antigen. So, see over here we are showing only one antigen. Okay. Now, in this one antigen, see there are 5 different epitopes. This is epitope 1, this is epitope 2, this is epitope 3, this is epitope 4, and this is epitope 5. Okay, now there are different B cells, multiple B cells, which will be producing the plasma cells, and thereby the antibodies, which will be coming and binding to the different epitopes to, elicit the process, to begin the process in the first place. Okay. Now coming to monoclonal antibodies versus polyclonal antibody. Okay. Now this is a polyclonal antibody because a same antibody is going and binding to different epitopes of the same antigen. And here comes the monoclonal antibody, which will be binding only to one region, one specific region of an antigen. Okay. Now, examples of monoclonal antibodies in the clinic I have given over here. So, for the first time, monoclonal antibodies were introduced in the year 1986. Okay, so this is a nice graph that I have given over here, which shows the cumulative number of human monoclonal antibodies entering clinical study between the year 1985 and 2008. Okay, so those line which are marked with yellow will indicate all human monoclonal antibodies. The red one will indicate only those human monoclonal antibodies which will be having anti-neoplastic effect, means anti-cancerous effect. Again, the greenish one will indicate immunomodulatory monoclonal antibodies only, means which will be involved in modulating immune response. Okay, which is again very important in different cancerous conditions and also in different diseases like HIV. Immunomodulation is very important. Then anti-infective monoclonal antibodies will be indicated by the blue ones. So see, these on the x-axis we have number of clinical candidates, and on the y-axis we have the years. Okay. Okay, now over 30 monoclonal antibodies are approved for clinical use by European and US regulatory agencies for a wide variety of indications, including but not limited to asthma, autoimmune diseases, oncology, and ophthalmic disorders. Ophthalmic disorders means different eye disorders, eye issues. Approximately 235 monoclonal antibodies are in active phase 3 trials for a wide variety of indications, including but not limited to Alzheimer's disease, which is a neurological disorder, autoimmune disease, cardiovascular disease, infectious disease, and osteoporosis, which is a very important and very common disorder, mainly in postmenopausal women. Okay. Okay, now this is a chart or table which explains the different cardiovascular monoclonal antibodies. Okay, so cardiovascular antibodies, monoclonal antibodies means they will be utilized for different cardiovascular problems, heart diseases. Okay, so on the left hand side we have MAB name, means the name of the monoclonal antibody. Then we have year, then we have indication for which disease it will be used, then different comments, and then the trade names. Okay. So MAB names, first one we have Muromonab CD3, which was discovered in the year 1986. Indication will be acute heart transplant rejection. Comment will be, it is the first monoclonal antibody to be approved. It is a murine monoclonal antibody targeting CD3 cells. Okay, murine means it's made from mice. Trade name will be orthoclone OKT3. Then we have amiximimab. It was discovered in the year 1995. Indication will be antiplatelet factor, used as antiplatelet factor. Comments will be chimeric FAB. Trade name will be Riopro. Again, chimeric means it's formed from different parts, utilizing different parts of different animals. Okay. Third one we have is digoxin immune FAB. Year was 2011, but only in United Kingdom, not in other countries. Indication will be digoxin toxicity. Digoxin is a medicine which is used, and it often is responsible for producing various toxic symptoms. Comment will be sheep FAB ovine. So it's made by utilizing FAB fragment collected from sheep. Trade name will be DGFAP and DGPIND. Okay. Now see, how will you discover a target for monoclonal antibody? Okay. So this is the signaling molecule, this is the receptor, this is the cell that we have. Inside the cell we have the nucleus where the DNA is there, and this cell is affected by the disease pathology. Let's consider. Okay. So the signaling molecule will be going and binding with the receptor. So the validated targets will be the receptor as well as the signaling molecule. Isn't it? Because we can only play or manipulate with these things if we want to free this cell from the disease pathology. Okay, we will have to modify or modulate the binding, the specific reaction between the signaling molecule and the receptor. Okay, now what is monoclonal antibody? See, mono means single and clonal means the ability of the antibody to go and bind with the antigen. That is the clonicity or clonality. So mono means single. So these antibodies will bind with the same epitope and they will be formed from a single B cell having genetically identical variable regions. So as we know, in the antibody there are constant regions, there are variable regions, both in the heavy chain and the light chain. Structure of antibody is something I've already taught, and in this session also I've given an overview of where the heavy chains are, where the light chains are, and where are the constant regions and the variable regions. So monoclonal antibody, the main though feature is, one is it will go and bind with the same epitope of the antigen. Second is it will be formed from a single B cell, single B lymphocyte when it is differentiated into a plasma cell from that. and hence, since it's formed from a single B cell, it will be having genetically identical variable regions. These are the two most important features of the monoclonal antibody that you need to remember. Okay, so as you can see, we have this mice that is there, and this is the receptor for the particular antibody. Okay, so this is the spleen of the mice. So we isolate the spleen, and from the spleen we'll get the B cells. Okay, because now the B cells are activated since the receptor has come. Okay, means the antigen has come, which is bind with the receptor. So the B cells have been activated. So now, along with the B cells, they will be forming the hybridoma cells. Now the hybridoma cells will be cultured in the particular culture petri plate under laboratory conditions. So from these hybridoma cells, see, we are developing millions and millions of monoclonal antibodies. Okay, and these monoclonal antibodies will go and bind with the single epitope on the antigen. So this is basically the antigen that was initially injected into the mice, as a result of which the B cells were activated in the spleen. We isolated the spleen, we took the B cells and we cultured the hybridoma cells. From there we got the monoclonal antibodies. Okay. Now how can we humanize these mouse antibodies? By genetically engineering the mice. So how will we do that? Scientists will use genetic engineering to create mice to contain a human antibody gene. Okay, how? So this is the mouse embryo that we have over here, which is containing the DNA or the nucleus. But what we are doing is we are silencing the mouse antibody gene. So this is the mouse antibody gene over here, which is silenced in the mouse embryo. And after, like, simultaneously we are doing two things. We are silencing the antibody gene, and along with that we are adding a human antibody gene inside the mouse embryo. So now the mouse has become an individual mouse. After that, what is happening, when injected with antigen, genetically engineered mice will produce fully human antibodies because its own gene has been silenced. Now only the human antibody gene inside it is on. So when the antibody is injected with the antigen, this is the antigen which is injected inside the mouse and inside the genetically engineered mouse. What will happen? Whatever antibody the mouse will form in response to the you know antigen will be a human antibody. So now we can easily use that antibody in the human body. Okay, now in this diagram we can see we have the selected monoclonal antibody gene which needs to be transferred to the Chinese hamster ovary cells for manufacturing. Okay, so this is the B cell over here, you can see. So the B cell is having obviously a gene of its own and the receptors on the surface which will go and bind to the particular epitope of the antigen. And over here we have the selected phage. Okay, so the phage means it's a bacteriophage, it's a virus which can easily go and infect a bacterial cell. Okay, and also, well, like on top of that, another important feature that it has is it can easily become integrated with the chromosome of the host cell. It can integrate itself with the chromosome of the host cell and start multiplying or replicating along with the host cell chromosome. Okay, so let's see what is happening over here. So we are isolating both the genes. We are taking out both the genes from the B cell as well as the selected phage. So after that, we are inserting this antibody gene into the Chinese hamster ovary cell for the manufacture, for upgrading the manufacturing process of the monoclonal antibody, because monoclonal antibody is already formed utilizing the selected phage. Okay, so see, after inserting this antibody gene into the Chinese hamster ovary cell, they are replicating and they are gradually increasing in number. So this is how this Chinese hamster ovary cell line has been established. It is abbreviated as CHO cell line. So it is established in the year 1957 by Dr. Buck at the University of Colorado, and it is the most widely used mammalian commercial production line. And but there are some viral cell lines which do not replicate in CHO. They include HIV, influenza, polio, herpes, and measles. So these are some of the viruses which will not replicate in CHO. So we cannot utilize the CHO cell line for replicating or for finding any solution, any monoclonal antibody against these viral cell lines. Okay, so this is how the CHO cell lines will look under the microscope, and this is how they will be generating the monoclonal antibodies inside the B cell. Okay, now coming to the upstreaming process. How will you upstream the therapeutic monoclonal antibody manufacturing process? Okay, so this is the cell that we have, that is the B cell that we have, and inside the B cell we have the chromosome, the DNA or the genetic material, as well as we have the different antibodies. So we take a stock culture from this B cell which will be containing 100 ml of the solution or the suspension. Then we do a small scale culture utilizing this stock culture. After that we take a small scale bioreactor. From there we go or we transform to a large scale bioreactor which can hold around 3000 liter, and the small scale bioreactor was holding I think around 1 liter or 1500 liter. And from there means we are gradually, what is upstream? Upstream means where we are gradually increasing the leveling of a particular process. So from 3000 liter then we will go to a industrial scale range operation which will be happening at around 10,000 liter. Okay, the small scale culture was around 1 liter and the small scale bioreactor was around 5 liters. Okay, and now coming to the downstreaming process. So how the downstreaming process will occur? Let's see. So these are again the different B cells which are containing the monoclonal antibodies. So from there we are just isolating the monoclonal antibodies only from the B cell, and then we are just inserting them into the column chromatography, which is HPLC. Okay. So from this column, through filtration obviously, from this column chromatography, we are having a formulation, filtration and fill up system. So basically it's industrial scale reactions, okay. And thus we get, after this column filtration and column chromatography, filling process, we go for therapeutic packaging, okay. So lastly coming to the conclusion. So these monoclonal antibodies are used for a wide variety of clinical indications. As I have already talked about different cardiovascular issues, different autoimmune disorders, okay. Then conditions like osteoporosis, cystic fibrosis, these monoclonal antibodies can be of huge help. So these therapeutic monoclonal antibodies can be selected by use of hybridoma technology, phage display or genetically engineered mice, as I already explained through the previous slides, okay. Humanized and fully human monoclonal antibodies have lower incidence of immunogenicity than mouse monoclonal antibodies. This is a very important point that I would like to talk about, because immunogenicity is something very important whenever we are talking about antibodies, isn't it? Because immunogenicity is, what is immunogenicity? It is the ability to give rise to an immune response, or it is the ability to elicit an immune response inside the body. So after injecting the monoclonal antibody, whatever monoclonal antibody it is, whether it is a chimeric one, whether it is derived from mice, whether it is partially human, whether it is fully human, irrespective of that, whatever monoclonal antibody we are utilizing, if it is failing to produce or if it is failing to elicit the immune response, then it is of no use. Okay. So that thing is very important, that considerable immunogenicity should be there inside that monoclonal antibody. That means the monoclonal antibody should have the considerable immunogenicity, okay. And once the monoclonal antibody is selected, the monoclonal antibody gene is transferred to the mammalian cells for manufacture, because for the upstream process, for for taking it up into an industrial scale, as I showed through column chromatography, filtration and etc., it's an industrial processing. It needs industry. It's not possible to be done in a lab. It needs an industrial setup. It needs a factory where the different infrastructure and machines will be there, okay. So that part is also very important, and for that what we need suitable cell lines like Chinese hamster cells and different other cell lines are there. But we need to, before choosing the cell line, we need to know whether our particular virus or our particular bacteria of interest will multiply or will replicate in that cell line or not. Because cell line means it's acting as a host cell, isn't it? And cell line we mostly need when we are culturing the viral cells, because bacteria or fungus, they can be cultured in cell in the laboratory only, just utilizing a culture medium, no need to give any cell. But for replicating or culturing viral cells, we need to give the cell lines, particular cells where that particular virus will go and replicate. Okay. So monoclonal antibodies can tag a cell for destruction, block cell receptors to interrupt disease pathology, or capture signaling molecules to interrupt disease pathology. So these are the various ways, the three major ways by which the monoclonal antibody can actually prevent or can actually treat or manage a particular pathological condition or a disease. So it will either tag a cell for destruction. So if, so for example, a cell has been infected by a particular virus, so if I inject that person with the monoclonal antibody, so it can directly go and tag that cell and tell the other cells of the body, or in fact tell the entire body that okay, this cell is infected, so you need to do something with this cell. Either you kill the cell or you save the other surrounding cells from getting killed by getting infected from that cell, anything that the body will decide. Secondly, it can block cell receptors to interrupt disease pathology, because whenever a disease is spreading inside a body, whenever a pathological process has started inside the body, it involves different cell signaling molecules and different cell signaling pathways. So if the monoclonal antibodies can go and capture and block those cell receptors or cell signaling molecules, then the disease pathological process can be halted or it can be stopped. Okay, so these are some of the the major part of this section is mostly this like explained by me the on conclusion slide only. So these are actually the main aspects that you need to know about monoclonal antibody. Okay, so monoclonal antibodies will be having a ability to bind with a single epitope on an antigen. They will be formed from a single B cell, and they will be having genetically identical variable regions. That's why they are known as monoclonal antibody, because clone means which is the exact genetical replica, and here the exact genetical replica will be found in the variable regions. Okay, and they can be utilized for various clinical purposes, for management of various diseases. And whenever you are coming up with a name which is a monoclonal antibody, always remember that it must end with the three words MAB, with the three letters MAB, which stands for monoclonal antibody. Okay, so I think that was a brief overview of protein therapeutics and the two sessions that I talked about. So we will be soon moving forward to the next topic or the next session of this course. Till then, thank you, goodbye. Please like, share and subscribe ITLS Academy. Thank you.